think we can get started now with the tutorial. I'm just going to give you like 2 minutes of introduction, and then there are some handouts over here which you can grab in a minute. So let me just tell you what the structure is here. We have two data sets. We have a triple TOF data set, triple TOF 6600, acquired using kind of the standard 64 variable window method that we use in our lab. And we have the same samples acquired on a QE, Q-Exactive Plus. And here the acquisition setup is basically following this diagnosis paper from 2015. So it's like I think it's 18 variable window method. So choose whichever one you like, or both if you're super psyched up. And the samples, so there's a handout for each of those, right? So you can take whichever handout you want, or both if you like. And the samples that we're going to be analyzing are from this paper that's been mentioned a few times, this Navarro LFQ bench paper. So here this is kind of a known it's a species mixture sample where we took E. coli, yeast, and human lysates and mixed them together in sort of predefined values. So the A sample has 20% E. coli, 15% yeast, and 65% human, and the B sample has 5% E. coli, 30% yeast, and 65% human. So the human proteins should basically be constant. The yeast proteins should be a ratio of 2:1 from B to A, and the E. coli proteins should be in a ratio of like 4:1 from A to B. So you know something in advance about what the ratios should look like, and the question is whether the data will actually look like that. So we acquired these data both in DIA mode using these acquisition methods with 3 technical replicates of each sample. And in addition to that, we acquired the data in DDA mode for 1 replicate of each of A and B. And the DDA data has been searched already magically for you using Comet and processed with TPP. And you're going to start from these search data results and build a spectral library. And yeah, maybe I should move to the next slide. So what we're actually going to ask you to do is basically comes in 2 sections. Number 1 is to set up the Skyline document for DIA analysis. So how we're going to set up the parameters and specify how we want to do the extraction of MS2 ion chromatograms and the precursor isolation scheme and so on. We're going to add these IRT peptide query parameters in and set up an IRT calculator. We're going to build the spectral libraries from the DDA data and add some target peptides just for like 10 or 12 example proteins to the document. So we're not going to extract all the proteins. We're just going to extract a few just in the interest of time to show you how it works. And we're going to add also decoys. So Skyline has a function for generating decoys. And then we're going to extract all that data. So section 2 is extracting the data. So we're going to import the DIA data into Skyline, which is going to extract the ion chromatograms. We're going to look at peak scoring and FDR control using this kind of mProfit type of algorithm. We're going to look a little bit at the data, hopefully look manually a little bit at the data. We're going to look at this mass accuracy and retention time prediction accuracy, whether this is within what we expect. Do some quantitative comparisons versus A and B using the built-in statistics in Skyline. And then last, we're going to look a little bit at the difference between the MS2 extracted data. Sorry, that should say MS1. Sorry, it's a difference between MS1 and MS2 XICs. And basically, the instructions are all written down for you there in these tutorials. So just grab the, and should be in this directory DIA LFQB. So just grab a handout and get started, and we'll be here if there are any questions.
Where's